I'm sorry.
Okay, well, good morning, everyone. My name is Mike O'Driscoll. I'm a hydrologist at East Carolina University, and I'm going to be moderating the session today for coastal water quality. And so we're going to try to keep on time. And our first speaker is Tom Vogel.
Hi, I'm Tom Vogel. I'm from East Carolina University, and today I'm presenting preliminary results from chapter 1 of my dissertation research. And it's using a spatial approach to look at nitrate groundwater contamination sources in the lower Cape Fear River. So just to quickly outline how this presentation will go, I'll start with a little background on the study area and the project itself, go into the methodology of my approach, some results and conclusions, and then future work related to my dissertation and this particular chapter. So just to start out, the study area for this project is the lower Cape Fear River Basin, particularly Duplin, Sampson, Bladen, Cumberland, Harnett, and Pender counties, all of which were severely impacted by Hurricane Florence, as well as Floyd, Matthew, and several others in the last 20 years. And these areas are predominantly, there's a heavy influence of agricultural and animal operation industries here. And this leads to several potential groundwater sources for nitrate. So just to highlight why nitrate is such an important contaminant in this area, agriculture is about a $73 billion industry in North Carolina, and about $8 billion of that comes directly from animal operations. The map on your left shows the points are all the animal operations in the eastern half of North Carolina, overlaid on the human population at the county level. And as you can see in the study area highlighted in the red box and excerpted so you could see the populations better, there is a significantly larger population of animals to humans. In particular, in Sampson County, it's about 36-1. And these animal operations represent about 55% of that $8 billion. So as I mentioned, this is about nitrate, and these areas are severely impacted by coastal hurricanes, where flooding is one of the major impacts. The floodwaters allow contaminants to enter directly into the groundwater wells rather than being filtered through the soil and the plants during infiltration. This can lead to direct contamination from fertilizers from row crops, as well as from the lagoon and waste storage structures for the animal operations, which were designed to withstand standard rain and precipitation events, but don't hold up to the changing weather patterns now, and in particular to these major storms that have come through in the last decades. Another potential hazard from the flooding is the inundation of septic systems and sewers, which, similar to when the lagoons overtop, release these contaminants directly into the surface or the water supply. And the pictures just show, the bottom shows a flooded hog operation in Sampson County during Hurricane Florence, and the top is just a house in the same area. And so in some preliminary analysis, we found that that homeowners in these areas are drilling their private groundwater supply wells deeper than they're required to, and we expected them to. We believe this is a preventative behavior to access safer, deeper groundwater. However, drilling deeper doesn't necessarily result in accessing a confined aquifer in this area. And so even though they're drilling deeper, if they haven't drilled deep enough to access one of the confined units, they aren't necessarily avoiding the health hazards of nitrate and bacterial contamination that they're trying to. And for the purposes of this study, all of the wells examined are in the superficial aquifer. So the research question that this chapter and this presentation is addressing is, where are the elevated nitrate contamination concentrations most likely to occur, and which upgradient land uses and sources contribute most to these elevated concentrations? One aspect of this project is that it's associated with an earlier EPA STAR grant, in which our team manually digitized and geocoded the GW1 forms for well drilling records. These are all hard copy in North Carolina right now. And this allowed us to get head data and geological data that it's publicly available, but it's not reported online. And so the wells examined in this are the ones that we manually digitized. The head elevation for these wells was determined by using the depth to water and then converting it to an elevation level using a DEM. And then the soil and geological features were used to determine hydrologic conductivity. So the map on the left shows you where all of these GW1 form wells are in the study area. And I used a kriging interpolation to determine the head surface here. That's because the wells are not measured at a uniform time. They're just measured when they're constructed. So rather than using an interpolation method that preserves the exact measurement at each point, I took one that better created a generalized consistent surface. Because these wells were all well within the study area, there were issues with the edge effects and catchment delineation later on. So I incorporated DWR monitoring wells, which very nicely surrounded and filled in the gaps that the GW1 data had. And this allowed a consistent head elevation surface to be created across the entire study area and facilitated analysis better. And again, both the DWR wells and the GW1 wells in this figure are all in the superficial aquifer in the study area. So as I mentioned, the head surface was created by a kriging interpolation method. A cell size of about 330 feet or 100 meters was used for the analysis. Initial trials at higher resolution data were not consistent, and the catchments created were not usable for analysis. They were too small. And during this analysis, there were 238 wells that were manually georeferenced and an additional 132 that are from the Division of Water Resources. So following the head surface generation, the catchments were delineated using the ArcGIS surface water hydrology tools, which is the reason that this study focuses on the superficial aquifer. At the deeper aquifers, there's no direct interaction between surface water, surface contaminants, and the groundwater. But in this one, it can be, the relationship is clear enough that it can be assumed that it will follow similar patterns, similar to the elevation that surface water follows. And the GW1 form wells were used as pour points. So each of these was the sink to which a catchment was delineated. From there, the animal operation and nitrate data was extracted into these. So the figure here shows the catchments as they were generated, and overlaid on that is the nitrate measurements that fell within these areas. There were about 428 of those. And from here, you can see about half of them, about half of these watersheds contain the CAFO operations. The larger ones are more likely to contain CAFOs. I believe this is partially a spatial effect. Larger surface area is more likely to capture. So once the data was extracted, it was exported as a CSV and the analysis took place in R. And the initial analysis I performed was comparing the nitrate concentrations across watersheds with regard to the presence or absence of a CAFO in the watersheds. And so the analysis determined that there was a significant difference among the watersheds and with regard to the presence of CAFOs. The figure on your left is a frequency diagram, cumulative frequency diagram for the two categories. The red line on top shows the control group, where you can see about 95% of the measurements were actually non-detects. They were below the detection limit of the state lab. From there, it rises up slowly. The blue line below it shows the frequency for the watersheds with the CAFOs in them. And it's closer to about 85% that had the non-detects, but overall the measurements were significantly higher in this area. I also, once that was determined, analyzed if the number of CAFOs present in a watershed had any significant impact on the nitrate concentrations. And while there was a difference between the presence or absence of an animal operation, there was no significant difference with regard to how many operations fell within a watershed on the concentration of nitrate in this area. So there are a few significant impacts, limitations on this study. The groundwater data is not uniform. The GW1 wells span several years and are single point measurements. So there's no consistent season or timing for these. The DWR wells are monitored over time, and for that purpose, they were just an arithmetic average was taken to turn what in some cases was thousands of measurements into one for this analysis. So some additional work on getting a more uniform method for that may be necessary. The other major caveat is that the nitrate data was predominantly non-detects. There were about 430 nitrate measurements within the catchments. 34 of them were above the 1 milligram per liter threshold. And only 7 of those violated the exceeded EPA's 10 milligram per liter recommended concentration. So some quick conclusions. As I just mentioned, the drinking water had a much lower percentage of samples contaminated by nitrate than we expected to find. So the water is cleaner and safer than we expected with regard to nitrate contamination. Based on this analysis, the use of GW1 well drilling permits to augment other groundwater data sources in these areas where there's sparse data to begin with can be viable for this analysis. And would potentially benefit future modeling efforts. And based on this, the spatial rather than numerical approach may be a viable method to assess pollution sources in these aquifers. At this stage, I'm not ready to identify sources using this yet, but it is an ongoing process for my dissertation research. And so some future work with regard to this model specifically, which is the first chapter of my dissertation, I'll be repeating the model to include additional land uses and contamination sources for nitrate, including agricultural land, and potentially industrial sources in the area where that data is available. I will also be incorporating hydraulic conductivity to better assess if the nitrate will be transferred more quickly through the areas. In addition, the second chapter of my dissertation will be a mailed notification study where I am trying to promote voluntary water quality testing in this area. And so while that focuses on bacterial contamination, it may also result in increased testing for nitrate contamination, and again, contribute to the available data in these sparsely populated areas where it would be beneficial. And then the final chapter of my dissertation is an integrated risk model, which will account for the physical variables in this one that contribute to groundwater contamination, as well as social and behavioral influences, such as people's willingness to test, that would be determined in chapter 2, to get an overall idea of how households are susceptible to contamination in their drinking water. And there are so many people that need to be acknowledged for this project. Randall, Jake, and Ariane are my advisors at ECU, as well as Kelsey Piper and Greg Howard, who are my other committee members. Rob Howard is an incredible GIS faculty member on campus that helps me a lot, and Sid is my program director. And then all of the people that were involved in the GW1 study and initial sampling efforts. And of course, ECU and the Coastal Resources Management Program for supporting me. Thank you. With that, I can take questions. Thank you.
Thanks, Tom. We have time for 1 or 2 questions if anyone has a question.
I not too familiar with the aspect of looking at nitrate. I was just wondering, have there been discussion on like whether or not it will add to your study if you also look at like nitrite or ammonia or other forms of nitrogen, just to see if there's a big difference?
There can be. Nitrite in particular is very small and not consistent in groundwater. It tends to convert to the nitrate. So it's usually represented in a combined measurement. Ammonium could be tested for. Nitrate is we chose nitrate because it's readily monitored for by the state lab and their inorganic testing panel. So it's the data that is frequently available on the public resources.
For your spatial approach, are you thinking about just like putting a buffer around the wells and like maybe changing the distance of that and looking at the number of CAFOs and land use in that?
I'm actually calculating all the catchments from those earlier maps. So that's why I'm using a spatial approach instead of, initially I tried a GMS numerical modeling approach on a much smaller scale to see if it worked. But the data was too sparse. So using the hydrology tool set, it's using a spatial and elevation-based approach where the head surface instead substitutes for DEM. And so then I'm using those catchments to complete the analysis instead. Does that make sense or answer the question kind of?
Thanks so much, everyone. I think we'll have to jump on to the next, which will be Natalie.
Good morning, everyone. This is my very first oral presentation ever, so I'm super excited and very much over caffeinated. So, hi, I'm Natalie Chazal. I'm in my first year of a master's program at NC State University, and I work at the Biosystems Analytics Lab with Dr. Natalie Nelson. So this past year, I've been focusing on utilizing fecal coliform observations from shellfish sanitation programs to analyze historical water quality trends in North Carolina. So As all of you know, water quality is a vital metric for monitoring the health of estuarine systems. And as it stands, compared to the tens of thousands of freshwater monitoring stations, coastal water quality is only monitored or has publicly available data through a couple hundred stations, despite this ecosystem's increasing importance. So even in North Carolina specifically, there's only 4 NERS dedicated water quality sampling stations in 2 designated reserves. And so we're all familiar with the variation and uniqueness of North Carolina's landscape. So we know that this is not nearly enough infrastructure to collect the data that we need to ensure the persistence of these ecosystems, which is where shellfish sanitation data comes in. So NCDEQ has been managing shellfish growing waters since the 1920s with a digitized database going back to the 70s. So you can tell that they have just a temporal and spatial extent that's to die for. So my project was to use this shellfish sanitation data to look at the historical water quality trends while also looking at the correlations in trends in other environmental and management variables. So to do this, we had 3 goals in mind, which were to assess whether we can actually use this monitoring data to infer anything about water quality, to analyze the fecal coliform concentrations over space and time, and to identify any possible management or environmental relationships to these fecal coliform trends that would indicate any drivers of change. So a bit more about the study system. So to manage shellfish aquaculture in North Carolina, NCDMS splits up the coast into smaller subdivisions called shellfish growing areas, or SGAs. So each SGA is labeled as restricted, approved, or conditionally managed. So these approved and conditionally managed areas are in green on the left. Sorry, I'm pointing in both places. And the water quality sampling stations that are used to monitor these areas are the little black dots. So the SGAs are coded alphanumerically. And so to subdivide our system into smaller, more ecologically similar areas, we use the letter at the beginning of that code to identify 9 major areas along the coast, as you can see in the middle map. So each conditionally managed area is assigned a rainfall threshold from 1 to 4 inches. And basically, fecal coliform loading in these systems is highly connected to runoff. So if a storm or rainfall event meets that threshold within a 24-hour period, that growing area automatically closes until the SGA can be resampled and sort of confirm safe fecal coliform levels. So yeah. Which brings us to the workflow. So how we use this management data was we took all of the routine sampling data from all of our stations that had routine samples. We filtered out the samples based on the continuity of the data within our time frame, which was 1999 to 2021. And I regret not putting that time frame on the slides, but that's what we're talking about, the past 20 years. And then we calculated Kendall's, tau, and send slopes for each of the stations. So man-Kendall testing is a commonly used test in environmental data to detect trends. And it provides us with a tau test statistic. However, that test statistic doesn't really give us an idea of the magnitude of the change, just whether there's a trend or not. So we use send slope estimation to give us an estimate of the magnitude of that change. So we have a send slope and a Kendall's tau. And just a heads up, I'll indicate send slope as beta, and I might accidentally say that too. But so we append all of those to a shapefile, and we can create maps like this. So basically, this map is showing each of the sampling stations along North Carolina's coast, colored by the tau value. So red and orange indicates increasing fecal coliform levels over the past 20 years, and blue and green indicates decreasing. And so each station is also sized by the absolute value of the send slope. So basically, the bigger the dot, the faster the change. And that inset map is just a little example of what this looks like in a little local area. And I chose my home county. So yeah. That was the first part of the study. So the second part of the study was to look for trends in environmental parameters. So the environmental parameters that we chose were land use and land cover, which was gathered, and the percent change was calculated for the 5 major land use types shown in that little table on the bottom. Salinity data, which was collected with the routine samples. So we basically just put that data set through the exact same workflow as the fecal coliform counts to get tau and send slopes for salinity. Precipitation data came from the North Carolina State Climate Office. There we go. And the trends in total precipitation or annual total precipitation were calculated. And then distance to shore is super straightforward. Just, the water quality sampling stations are spatially fixed. So that was just a nearest calculation. So with all of that, we calculated a correlation matrix with the Pearson's correlation coefficients. So here are the results of the analysis between the variables aggregated along the full coast of North Carolina. This is just a wall of numbers, but some results to note here are the clear negative relationships between fecal coliform trends and salinity trends. And this is expected based on what we know about fecal coliform dynamics in estuarine systems. And the weaker positive relationship between fecal coliform beta and precipitation trends that we're seeing, which are also expected just based off of what we know about increased precipitation causing more runoff, which means more fecal coliform loading in the system. And there are a couple more interesting relationships to note, but I always go over time with this presentation. So in the interest of time, I'd like to move on to the trends broken down into those finer spatial scales. So this chart is basically showing the Pearson's correlation coefficient for each of the relationships between fecal coliform sen and land use cover categories on the left and other environmental variables on the right. So I'm going over. So I seriously urge you to go back and look at this and ask me questions or email me. But what we're going to focus on right now is salinity. So basically, this plot on the left is a histogram of the salinity beta values for each of the growing areas. So also warn you about this figure. SGA is the southernmost and H is the northernmost, as you can see on the map. Just wanted to give you a heads up. But yeah, so here we can see that there's a steadily increasing salinity beta value as you go from A to F. And this increase in center is also met with somewhat narrowing spread. And so this trend breaks down after G and H, where we sort of see that increase in spread again and the decrease in salinity beta values. So yeah, it makes a bit more sense later on, but you can see in certain areas, the salinity samples that DMF are getting are showing an increase in salinity in certain areas, especially SGA, D, E, and F. So on this next slide, SGAE in particular, where we saw, these really high salinity beta values, we see that there's a cluster of stations showing improving water quality as well. So that sort of blue cluster right in the middle, not the big dot, but the little, Carteret County area. So this area in particular is sort of tricky to interpret because of the known degradation in water quality that contradicts these decreasing fecal coliform trends. But if we look at the salinity results for this area, we can see that these samples are being taken at times where there are higher and higher salinity levels. So this leads us to believe that salinity could sort of be an indicator of the conditions that NCDMF is taking the samples in these particular areas, and could sort of be an indicator as to the extent of bias or sampling bias when you're looking at these localized trends and trying to interpret water quality trends from sanitation data. So to try and capture these local nuances in the interpretation of these fecal coliform trends in relation to their environmental parameters, we chose 3 focal areas to focus in on to sort of get a representative sample of the different ecological and maricultural landscapes. So we define these areas based off of estuary type, rainfall thresholds, developed land use, which is shaded in purple, and leasing, shellfish leasing activity. And so all of those are sort of labeled on the left. So you can see B at the bottom, there's a generally increasing fecal coliform trend, especially in the southernmost area, near the Carolina Beach and Bald Head Island locations, where there are more decreasing fecal coliform trends in the northern lagoonal areas. So basically, we think that's just because of this highly, highly developed Wilmington area, sort of degrading water quality moving southward down the Cape Fear River. And so this relationship between increasing fecal coliform concentrations and associated with high intensity development, it tracks with what we know or what we would expect from a system like this. Focal area E, as I said before, it is also experiencing not as much development as Wilmington, but I mean, still moderate development to a degree, which sort of, sorry, refocus. Mild development, which we would expect to get decreasing or increasing fecal coliforms, but we're seeing decreasing fecal coliforms. So if you remember back, this area also had the highest salinity beta values out of all the other growing areas and was associated with the only positive relationship between those two. So the difference between B and E is also seen in the rainfall thresholds that are assigned to these areas. So B has relatively high rainfall thresholds, so 2.5 to 4 inches in that area. And then E has very low rainfall thresholds, so like 1 inch. And so we think that because routine sampling can only happen when areas are open, and because areas within E, because they have such low rainfall thresholds, will be closed more often, the samples that are being taken for E are at times where you would be expecting more, expecting better water quality samples or lower fecal coliform estimates. So yeah, so area G, on the other hand, has very, very high, that's all 4-inch rainfall thresholds. So basically, we think that this area may be more representative of the true water quality dynamics, just based off of the samples, because the samples are being captured at a more diverse range of conditions. Yeah, so with all of that, the main takeaways from this that we've gotten so far is that shellfish sanitation data can be used for long-term water quality inference with lesser concern for the sampling bias in areas that are not conditionally managed, or conditionally managed, especially with low rainfall thresholds. But even then, results from the conditionally managed areas can demonstrate the effectiveness of current and past management decisions in particular areas. And then the trends in salinity can provide further insight into the sampling bias from the sanitation data for particular areas. And then focal areas B, E, and G can sort of be used as case studies for interpreting these results in different, under different management landscapes and different environmental parameters. So, any questions?
Thanks, Natalie. Do you have any questions for Natalie?
Back in the 90s, Dr. Nancy White led a study in Jumping Run Creek in Carteret County. And it was a time series analysis of looking at aerial photos to the fecal coliform data from DEQ. And she found that removal of forests combined with length of ditches were the best predictors of the change in fecal coliform. So that's just the FYI.
That is really interesting. Yeah, that'd be fantastic. I'll get your email afterwards.
It's a great presentation. Do you think that all this data can inform the shellfish sanitation approaches to closures?
I really hope so. And our lab is doing a lot of really exciting work with the shellfish sanitation data, especially because I only use the routine sampling, but there's conditional sampling that could also be super informative. And I know that Megan Carr in my lab has developed a decision-making tool that sort of leverages machine learning approaches to predict when or to sort of estimate when fecal coliforms will be under the NSSP threshold after a certain amount of time. So I think that this work is really exciting, and yeah, it could be used for more decision-making tools like that.
Hi, I'm just curious about, do you know the process that they use to collect these samples? Is it water samples or is it like samples from the actual shellfish?
So they do take meat samples, but I am using the water quality samples. So I should know more about this, but I think they're grab samples. I come from an animal science background, not a water quality. So I'm not really sure what that means, but that's what I've been told.
Okay, I get, thank you.
I just wanted to say I loved your graphics. They were so very nice.
Thank you so much. I made them myself. Oh, that is so great to hear. Thank you.
Any other questions? We've got a couple more minutes. I don't see any questions online. Nathan, and then you can get set up for your talk.
That's right. Did I understand that I was a little confused about the increases in salinity in Carteret County being a bias, or how? Yeah.
Yes. So basically, these samples are being taken for day-to-day management decisions. And so the purpose of this study, they're taken for day-to-day management decisions. And so they're not necessarily taken systematically in a way to inform historical analyses like this. So basically what I'm saying with the salinity stuff is salinity could be an indicator of the conditions that they're taking the samples in, which doesn't could sort of help you interpret the results from the analysis, if that makes sense.
So do you believe there's been a salinity increase, or is that?
Yeah, so I actually don't know. And there's no salinity or unbiased salinity data to sort of ground truth this with. So that would be on my wish list. It would be a salinity time series, especially for Carteret County. But yeah, thank you for that question.
Any other questions? Yeah, Nathan, you have to come up too for your talk. So. Gloria.
Thank you for the great presentation. Thank you for the claps for my question. Natalie, I was curious, you considered land use cover changes. I was curious if you considered the presence of stormwater ponds, that we have so many regulated stormwater ponds along the coast.
I have not, but that would be very interesting to look at. I'll get your email and I'd love to follow up.
Thanks so much, Natalie. Next up is Nathan Hall.
Much better. Try to keep it up to my face so I can talk in the microphone. Thanks everybody for being here. I'm going to talk about a project that's I've been working on in collaboration with APNEP to look at water clarity thresholds for protecting and restoring submerged aquatic vegetation and within the Albemarle-Pamlico estuarine system. I think we all know that seagrasses are really important. They're foundational species. They're critical for fisheries habitats. They stabilize sediment. They store carbon. They do a lot of things we like. And unfortunately, APNEP's been surveying them for, I don't know, 15 years or so. That's not the unfortunate part. The unfortunate part is through these surveys, they've noted some pretty bad looking declines in SAV, both in our high salinity waters, particularly in Bogue Sound, but also, and especially in our lower salinity waters. And particularly in that case, in the Albemarle Sound region. So there's reason to be concerned about our seagrasses. That's kind of where this project came about, was to look at our current water clarity standards and to see how they stack up against what we think they should be to maintain healthy seagrasses. Trying to right click or left click. Right. Okay, it wasn't me. Can I just use the keypad? Yeah. Okay, I'll do that. Cool. Well, right arrow. All right, I broke it. I want some minutes back. Okay, well, I'll just keep going. We know what light requirements our seagrasses need. I should mention we have two different types of seagrass habitats in North Carolina. We have high salinity seagrass meadows. And these species they don't grow high up in the water column. They stay right along the bottom. Okay, great. And they are out here in this high salinity areas back behind the Outer Banks. That's most of our seagrass area in North Carolina, is these high salinity seagrass meadows. We also have low salinity seagrass species that form canopies. So they grow up from the bottom and they kind of lay over at the water surface. And in doing that, they're exposed to a lot of light. So the water clarity isn't as critical. So they don't need as much as clear of water. So we know this mostly from studies that have been done up in Chesapeake Bay. So we've adopted the water clarity thresholds that they need from the studies that have been done up in Chesapeake Bay. And what it amounts to is, so when light comes in, hits the surface, it declines exponentially through the water column. And you can describe that exponential decrease with a K value, that exponential equation. And so for the low salinity species, we know they need about 13% of incident irradiance down to about 1.5 meters deep to get the seagrass acreage we want in North Carolina. For the high salinity species, we need about 22% down to about 1.7 meters to get the seagrass acreage that we want. And that works out to these K values. For high salinity values, about 0.89, and for low salinity, it's about 1.36. So this was kind of established before I even came into this project. My goal for this project was then to figure out, how do we get to those light attenuation coefficients, or how do we stay there in cases where we're already there? So there's three things that govern light attenuation in our estuarine waters. There's the suspended sediments, and we can measure that using things like turbidity. There's the amount of algae that's in the water. We measure that with chlorophyll A, typically. And then there's also color dissolved organic matter, CDOM for short, and that's the T color of the water. And in some places, that can be the most important thing attenuating light, particularly in some of our coastal streams, like Pasquotank River, for example. It's coffee water. It's really dark. You have to understand that background T color if you're going to try to predict what levels you could let chlorophyll or turbidity come up to, because that background attenuation strongly affects what those chlorophyll or turbidity thresholds can be and still meet your what you want to see get into the plants. So this study uses an optical model to calculate what that K value is going to be with different levels of chlorophyll, turbidity, and a background of CDOM. And importantly, so you can feed the model those three things, that's going to tell you what the K value is. You can also use the model in reverse and ask the question, given that we want to be here at this level of light attenuation, what values of turbidity and chlorophyll can we have given a background of CDOM, which we can't control at all? That's completely natural. We can't, there's no management strategy really to change CDOM very much. So given this background, what levels of chlorophyll and turbidity can we have and still achieve the light attenuation levels we want for seagrasses? So that's what's shown here on this graph. This dark red, or this red line, is a line of constant light attenuation where seagrasses can persist. Anything lower than that line, seagrasses are really happy. Anything above that line, seagrasses will fade away. And that line can move up and down given different levels of background levels of CDOM. So if you look at your average conditions in estuary and your chlorophyll and turbidity put you above this line, you would like to come back down to that line where seagrasses can persist. And you could take lots of different ways to get there. You come straight down, kind of like reducing chlorophyll a little bit, reducing turbidity a little bit, come back down the line, or you could assume maybe you don't think you can control turbidity, but you can control chlorophyll. So you come straight down or straight across in this case to come back to that line. So this can tell you how you need to manage sediment inputs or nutrient inputs to achieve the turbidity and chlorophyll that you want. So the goals of this modeling project were to determine what the chlorophyll A and turbidity needs to be in low and high salinity waters to meet the seagrass requirements, and then to compare that against the current state standards and see how they match up. Fortunately, so this is using an optical model, and we already have an optical model. There was an optical model that was calibrated in the North River. So it's within the Albemarle-Pamlico system, but it's on the southern end, and it's not really that strongly hydrologically connected to the rest of the Albemarle-Pamlico system. So before we could just set out and use this model, the first step was to validate the model for use in other areas within the Albemarle system. Then once we got the model validated, then we could use the model to assess what chlorophyll and turbidity levels need to be and how that stacks up against our current water quality standards. So for high salinity areas, the model worked pretty well. We had, these are plots showing different areas, the high salinity waters where I had data available, meaning I had CDOM information, I had turbidity and chlorophyll information to feed the model to predict that light attenuation value. And then I compared that against measured light attenuation. So that's predicted from the model on the x-axis, observed on the y. And for the high salinity, even though there's a lot of scatter, there's fairly low bias, and it's pretty reasonable. So this is, I felt pretty good about this. For low salinity estuarine waters though, the model severely underpredicted light attenuation. So there's something different optically about probably in the particulate fraction in the low salinity waters that's making this model not work very well. And so I wouldn't feel comfortable trying to use this model to go to the next step in low salinity waters and predict what those chlorophyll and turbidity values need to be, because the model is going to say they need to be, that they can be a lot higher than they actually should be. So for the next step, I'm not going to talk about the low salinity stuff anymore, because right now we're working on recalibrating the model for low salinity waters. But in the meantime, I'll talk about the high salinity chlorophyll and turbidity thresholds. So each one of these plots shows all the turbidity and chlorophyll data that we have. And the big yellow dot is the median value. That median value is then used, that's the average condition of both turbidity and chlorophyll. That median value is then either drawn straight across or straight up to determine at what level of chlorophyll or turbidity intersects with that line of constant attenuation, which is that amount of attenuation that seagrass that we need for maintaining our high salinity seagrasses. Doing all this, we come up with a bunch of different turbidity and chlorophyll thresholds for these different water bodies in our high salinity estuarine waters. And you can just kind of look around the values. The turbidity values range from like 3 or 4 to 9. Chlorophyll values range from 3 to 33, I think. The higher chlorophyll value thresholds there are the ones that are real close to the inlet. They have almost 0 CDOM, so there's none of that background attenuation. And they also have really pretty low turbidity values a lot of times too. So it's that's like the extreme case. But for the most part, our chlorophyll thresholds kind of center around that 10 to 15 microgram per liter range. And for turbidity, it's somewhere around 5. So those are kind of the thresholds I think that we can justify saying we need for our high salinity seagrass habitats. The next tricky part is, so these thresholds are based on median values. Half the time it's higher, half the time it's lower. State standards for chlorophyll and turbidity are do not exceed. So they say it shouldn't be higher than 40 micrograms per liter of chlorophyll A. But they allow for a 10% of samples to exceed that 40 micrograms per liter for just natural causes. You know, maybe there's a flood or a drought that somehow naturally creates conditions for blooms to develop. So in effect, the thresholds that I've developed are for a median value or a 50% quantile, and the state standard is being assessed at the 90% quantile. So you can't directly compare these two things. It's completely apples to oranges. So to get around this problem, what I did, and this is where me trying to explain this even to myself gets a little tricky. Oh, and I should mention one more thing. Not only is it assessed at 90% quantile, we have to have 90% statistical confidence that more than 10% of samples has exceeded the standard before it can be put on the 303D list as an impaired water body. So with that statistical confidence, you actually have to have quite a bit more than 10% of the samples exceeding 40 micrograms per liter for chlorophyll before it's going to be listed as impaired. Normally with the state sampling monthly and using 2 years of data for the assessments, they have like 24 samples maybe. You need almost 20% of samples to exceed the standard before it can be assessed. And it's totally based just on sample number as to what that percentage has above 40 has to be before it would be called impaired. So what I've done is I've gone through all the different stations within estuarine waters in North Carolina and looked at that, the number of samples that would be required, or the quantile, the critical quantile, which you would have to have that many samples exceed before it would be called impaired. And I looked across all these different stations that have all kinds of different distributions of chlorophyll A. I mean, different like average levels. And then compared that against the median values at those stations. So this is a way to say at the number of stations or the number of samples that would be required to be exceeded, how does that compare to the median value? And this is the apples to apples comparison. So what we can see is at the current standard of 40 micrograms per liter, that corresponds in North Carolina estuarine waters, the median value of about 20, which is a lot higher than those thresholds that we identified using the biooptical model that were like in that 10 to 15 range. So our current standard is not, and the way that it's assessed, and it's really hard to separate the standard from the way it's being assessed, because in effect, the assessment methodology determines how effective the concentrations relate to what's being impaired or being determined as impaired. Anyway, currently state standard is higher than these thresholds, so it's not being protective. And the same situation is true for turbidity. A 25 NTU standard with the way it's currently being assessed is, more than double what we really need to see to protect our high salinity seagrasses. How about 5 minutes? Oh man, tons of time. I'm almost done. There's gonna be lots of times for questions then. Okay. So that kind of wrapped up that part. But this is looking at using all of the water clarity data that I had available, which was a whole lot of Secudis data that I had to use empirical models then to convert to PAR attenuation. So this is a whole bunch of different Secudis data, some real PAR attenuation data for the Neuse River and Bogue Sound. We had some PAR attenuation data. The EPA measures in their coastal condition assessment, they measure PAR attenuation. So we had some actual PAR measurements, but a lot of this is DMF data from their trawl surveys from Secudis data. A really useful and widespread, spatially expansive data set of water clarity data. What I did was I compared that amount of light that both the high and low salinity seagrass meadows need to what's actually there. So that's what's shown up in here with different colors. The colors, if it's this 2x, means that there's twice as much light getting to the bottom that the seagrass needs. So that's excellent. And the only place you see that is Core Sound. Green is good. Green is like got 100% of what they need. And you see some of that in the lower parts of the estuary, low salinity estuaries. You'll see that those lower parts of the Neuse River currently light attenuation is good for seagrass. But you see an awful lot of red, particularly up in Albemarle Sound, along the inner banks. Yeah, and even in our outer banks areas where most of our seagrass meadows are, or the largest acreage, we're in that fair range for most of these stations. So I think there is reason to be concerned. Okay, so I'll just wrap it up. We saw that the chlorophyll and turbidity thresholds that are good for SAV aren't currently being protected by state water clarity standards. So we have some work to do there. And this is something that the nutrient criteria development plan is using this information to work on this. Currently, water clarity in a lot of the areas isn't meeting those clarity standards, or that what we think that the seagrasses need. And then in continuing work, like I said, we're recalibrating the biooptical model for the low salinity waters. So we'll have a much better ability to calculate these thresholds for those once the models reproduce. And we're also collecting, one of the hard parts about this project was CDOM data is very sparse. So a lot of the CDOM data that I had to be estimated using empirical models with salinity and a few other things. So, I mean, the models worked okay, but we're measuring CDOM a lot more expansively now, so that'll help too. That's it. I'm happy to take questions from the minute I have left.
Thanks so much, Nathan. We have time for one quick question if anyone has a question for Nathan.
Thanks for that presentation. So my question is about your second to last slide, where you showed the estimated and the measured.
Yes.
So the one after that. So it looks as though in the Albemarle, a bunch of the measured values are good or fair, but the estimated values are orange and red or terrible. Is there a reason why those would be different?
There is, and actually it's hard to see, but those numbers that are up there have to do a sample size, and those ones that were measured were the ones from the coastal condition assessment, and it's like 2 data points. So that I think they just went out on a nice clear when it was like hadn't been windy for a while, or yeah. I don't take a lot of stock, and if you look across the whole region, a lot of those measured values are some of the worst, like poorest clarity too. So I think it's just a sample number thing. I don't have a lot of faith in those, one sample size, N equals 1.
See me. Next up, we have Astrid Schnetzer.
Hello, everybody. I couldn't have asked for a better talk before mine. This was a wonderful introduction to cyanobacteria, wonderful pictures of the critters, which always get a bad rap, but they're really important for our ecosystems at the base of the food web. And I think I can go really quickly through my introduction because Amy has covered a lot of these things. So I want to talk mainly about a community collaborative research grant that we got funded through Sea Grant, where we try to look at where the toxin is going, specifically in the food web. Before that, I think I can go through this pretty quickly now because we've heard about this. In context, there is a general notion that these HAP events and their toxicity is increasing in varying regions. So we have certain concerns about this. There's a clear indication that eutrophication obviously plays a role, and how that happens, what nutrients and which ranges can favor certain species which are associated with certain issues. There's also an increasing amount of literature that tries to pair how these conditions might be tied in with climate change. And there's a lot of reviews that are now indicating that if you have a system that is already heavily nutrient loaded, that climate change could exacerbate what's going on with HAPs. So it depends on these obviously multiple factors. What I want to talk about is cyanobacteria, which you've already heard about. Cyanobacteria or blue-green algae are freshwater and estuarine issues. You see a little picture here from the Chowan River. And I'm also talking about 2019, like Amy did. It was a really high active bloom year in that region. I think this is also something we can skip through really quickly. Some of the issues and concerns come from excessive biomass alone, but then also can be tied in with if these critters that are blooming produce toxins. And there's all sorts of indications and studies that show that the harm that is done across the US or globally can be in the millions to billions. And that comes from all sorts of trickle effects here, from water quality to tourism to property values. So this is really a huge issue that ties into all sorts of issues for communities. Quick slide here taken from Sung et al. So how do we look about toxin exposure and potential pathways? You have in the middle your algal blooms, and in the beginning of a bloom when it's thriving, you have most of the toxins typically contained inside the cells. And that is an issue in regard to organisms within the water column in the system, filtering and accumulating these toxins inside the algae. And you can see a couple of examples that have tested positive for algal toxins within the food web. And then as these blooms dissipate, you can also find that these toxins are released into the water. And then these dissolved toxins are a concern in regard to recreational use or water drinking water sources. There's also an increasing amount of research. And for those of you who were here yesterday, Dr. Pearl talked a little bit about some efforts on trying to figure out how these cyanotoxins can also be aerosolized and cause sickness and concerns. Our lungs are really good filters, so that's could potentially be an issue in areas where that are really prone to those toxic blooms. And obviously also what you're seeing here is that some of the fish and benthic organisms also might have the potential to accumulate these toxins, and then we're in big trouble. All right, one thing that Amy also mentioned and that we're finding, and there's a lot of research on the West Coast, is we really have to take a closer look at where these toxins are going. Some of them seem to have very long residence times, so we don't just find them in the areas where those blooms occur, but all the way into the coastal systems where they might merge with other issues or other pollutants. This is, I hope it works. Let's see. Did I do this? There we go. This is a little video from the Chowan River in 2019. And I should say this is from Charlton Godwin, who's my collaborator on this grant. showing how it can become really tricky following these blooms and documenting them properly. And I'm gonna go through this list pretty quickly. These events can come and go really quick. They're very patchy, as this video shows. We've heard before, not all species are toxic. So even if you see a lot of cyanobacteria in the water or a lot of chlorophyll, which is a proxy for algal biomass, that doesn't equate to toxin, fortunately. Even if you have toxic species, they might actually turn it off and on, depending on who we're talking about. And then it almost seems like these critters want to make it hard for us, but they're just really diverse. Some of them can produce multiple toxins. When we have those multiple toxins in the system, we're just starting to really figure that out and catalog that quantitatively and qualitatively, which is really important. And then obviously, which toxins are there and which ranges should inform some of the exposure risk approaches, assessments, and guidelines. And there's I think I have the reference later, there's a really good new book by Kors and Welker, Cyanobacteria in Water, that goes through in detail how these guideline values are created and what we know and what we don't know. So I actually look there a lot because it's not easy to wrap your head around how these threshold values are created, and that typically come from mice studies. And then, which brings me back to the data, I want to introduce you to the fate of the toxins. Where's it going? Does it stay in the dissolved phase? Does it go into the food web? Is it aerosolized? And we looked at the food web. A quick plug for my lab real quick. This is different locations where over the last couple of years, we've looked for cyanotoxins. All the locations in greenish color are typically water treatment reservoirs where we've looked over 1.5 years. The yellow ones are places where we had typically summer studies. And then with the white ones towards the right, these are studies where we're now looking at the next trophic level and where the toxins are going in the food web. So a lot of data, and I probably left some people out and some funding sources. I apologize if it did. Most of this, and we heard this from Amy before, is due to people alerting us. We're not in the environment. We don't know when things are going on. And we had really, really wonderful collaborators, citizens, non-profit groups that told us, if you're in the area, come up here. It's scummy right here. Come here and sample. What are some of the main takeaways from all of these different studies? Multiple cyanotoxins are found year-round, not just in summer and in fall. If I say multiple cyanotoxins, we're regularly looking for microcystin, anotoxin, cylindrospermopsin, BMAA, and saxitoxin. The first four are typically there. Saxitoxin is the good news we hardly ever find. And if you want more information, by all means, just let me know. They occurred simultaneously, so there are some questions about how could these mixtures of toxins be an issue. The good news is we typically find them in very low concentrations, not even coming close to some of the drinking water thresholds, but there is exceptions. What else? Yes, and then we also find them, the cyanotoxins making it all the way to the like Bogue Sound, Kings Bluff, and Core Sound. We have some sampling, sorry, not Kings Bluff, Bogue Sound, Core Sound, and further down in Masonboro Sound, where we see some of the marine toxins, domoic acid that we're looking for, which is the cause of amnesic shellfish poisoning, merged with microcystin. So we're paying attention to this and trying to figure out how these distributions work over time and space. Huge plug for my students. They have several posters that look at some of these studies here, and Kirsten looks at cylindrospermopsin in Lake Matamosquite food web. Will has presented some of the data with the UNC collaboratory that we're collecting in Falls Lake, and Maddie can tell you some things about those merging toxins in coastal environments. All right, time to get to the study. So the CCRG grant came on the in the follow-up of issues in the Joan River in the Western Albemarle 2019 was a very strong active bloom year. There were at least 13 blooms that were reported within the region by DEQ. And there was a really, really big amount. There was a high concentrations of microcystin that we detected at varying bloom events throughout the Jordan River upstream, which is about 30 miles from where we conducted this food web study. And we actually separated the scum from the water column and found values within the scum over 1,000 micrograms per liter, which is 2 orders of magnitude above the recreational threshold. So yeah, don't let your dogs drink any of that water. Please check out Emily's poster. She's actually looking at these bloom events. She's dissecting them not just in regard to microcystin congeners, but she's also looking at degradation of these blooms and species composition for them. So she has a wonderful poster. All right, so what's the fate of the food web and how did we go about this? So we collaborated with Charlton Goodwin's group, and they do a ton of surveys out there. So again, a wonderful opportunity to collaborate. And we went out on weekly to monthly sampling events, and you can see that we got a variety of fish and in some cases blue crab through gill netting, seining, and trawling. And then I want to point out that this was done over weekly to monthly sampling for May till October. One of those places we revisited regularly, and this is Emily harvesting some Rangia clam, where you see the red circle. This is a place where we went back every time when we went out for the other sampling to see what is going on at that particular place. So it's kind of our control site where we also sampled in the water. All right. I'm not sure if Charlton is here. I'm not a fish person. If you have any fish questions, you have to talk to him. These are some of the stars of the study. We did a total of samples with 234. The Rangia clams were collected regularly throughout the study. And we also looked at different types of samples whenever we could. Some of the juveniles were too small and they were grinded up entirely. What were our targets? We were looking for microcystin and cylindrospermopsin. The reason for that was that we had some in situ tracking data from the area, and we found that those were the most prominent toxins in the water. You can see here, there's a lot of things listed here. I won't go through detail, but the one thing to take away is there's certainly a concern in regard to what they do to us if we ingest them. So here's a couple of acute health effects in humans, and then here is the levels through the World Health Organization that are referred to as where you have to cut consumption. And again, I mentioned Coors and Welka, they really nicely explain step by step how you get these values. What I've listed here, the tolerable daily intake for with 0.04 or 0.03 for those two toxins, so MCI is microcystin, and Psilus cylindrospermopsin, is estimated for an adult with 75 kilos. So what you're seeing here is that if over the lifetime of this adult, you could get up to 0.04 micrograms of microcystin on a daily basis. The lower value, the NOEL, no observed adverse effect level, is also in this case for the same, an adult with 75 kilos. And this is what you would take in one setting, one meal in one day. So this is where it becomes a problem, where you could start to see symptoms if you exceed that. All right, finally, we get to the data. All right, so let me walk you through this. So on the y-axis, you see micrograms toxin per kilogram animal tissue. This is in a log scale because the differences between where you look in an animal is pretty big. You can see that gut, viscera, and liver are way higher, about an order of magnitude. This is not new. People have found this over and over again. And the good news is that once you get to the muscle, which is the black bars, the amount of toxin drops pretty strongly. So this is good news for people who are consuming these animals, including me. When you see those little asterisks, this is when we didn't collect data. If you don't see a column represented, for example, for the mussel sample for the blue catfish in the beginning, that means we couldn't find microcystin in muscle tissue. So it was below the detection level. So good news here. We're now looking further into, and this is where Charlton comes in, into the ecological background, the feeding ecology of these organisms, to tie that into how they might get to the toxin, whether they're planktivorous or benthic feeders. So this is microcystin. Also in here are the levels that I said before with NOEL and TDI values and where we fall. And generally, I think we're in a good place, except if people eat these tissues that might be really heavily loaded, which I don't think is the case. For the Cylindrospermopsin, you can see that there are some species who also have these, the second toxin, but you see it way less frequently. And also I want to draw your attention that we're now talking about a whole order of magnitude lower levels. So microcystin is certainly the most common and higher in concentrations. All right. Another thing that we did is I told you we also did whole fish. So you find here, these guys were 2 to 4 centimeters in size that we grounded up entirely. And you can see that we have consistently high levels. This is certainly tied into feeding ecology. These little guys are filtering directly out of the water column, and that's how they get directly to the algae. 3 minutes, okay. Also in here is the average and standard deviation for the clams all the way to the right-hand side. And what I did here is because the value of micrograms toxin per kilogram animals is a little hard to comprehend. I looked up what the average meal of a fish fillet is, which is 150 grams. And what you see in the diamonds right there is what you would get if you ate 150 grams of each of these organisms and where you would fall in regard to what you would take in or consume. So good news, a little bit over if you eat it every day, but other than that, you're in a good place. The final graph I want to share is now a little bit of a temporal resolution here. So this is, let me walk you through this. This is the clam bed, and this is the triangles in yellow show you the tissue load in the clams. These are averages, and they're represented on the secondary y-axis as clam, micrograms, microcystin per kilogram clam. And it's laid over the chlorophyll, which is the green area. It's also on the secondary y-axis with micrograms chlorophyll per liter. And then compared to the toxin in the water column or the algae themselves on the primary y-axis. Sorry for this, it's not that complicated, but what you see in the squares, in the blue squares, is what we measured in the water compared to the triangle, triangles, which is in the shellfish. And there's a couple of things to take away from this. So chlorophyll, as we've heard before, does not equate toxin. This is just another way to look at that. But then also what we find in the water column surrounding those clams is not indicative of what's going on inside the clam. The clam can be toxified and you won't know it if you just sample the water around it. Yes, down to 1 minute. So next steps. We're looking at the environmental data we have for this. We're also looking at the feeding ecology of the species that we analyzed. We're gonna dig into that and see where those toxins are likely coming from. There's something to say about the blue crab and the clams, which are benthic feeders, and them having Cylindrospermopsin together with some of the fish while others don't. We're gonna look further into how filter feeders might be a good sentinel. This is not a new idea. This is being done on the West Coast and then up north. So this is a good idea probably for these environments. And we're also digging into the cyanobacterial species that are actually being ingested and are available in the water. And with that, I want to thank all the brilliant students. Some of them are here in the lab that are doing all the hard work that I get to present and be very proud of. Thank you very much.
Thanks so much, Astrid. Sorry, we don't have enough time for questions right now, but we will have a break right after this. So next up is Randall Etheridge.
Thanks everybody for sticking around. I'm in the coastal water quality section, but if you're here to hear about water quality, you're going to be disappointed. I think they didn't know where to put me, so they put me here. I do have the coastal part though, so you're going to hear about coastal stuff. So I'm just going to start by thanking my collaborators. You'll see this is a pretty interdisciplinary project. My background's biological and agricultural engineer, and Raymond Smith's an industrial engineer. Linda Deanna and Cindy Grace McCaskey are both social scientists. And JT Philho, the last collaborator, is an electrical engineer. So we had quite a few folks working on this. My background is in water quality, but as you'll see today, my research agenda has more been driven by the communities I've been working with than necessarily what I was most interested in. So ECU is in a region where a lot of the counties are considered economically distressed. And we took this into account thinking they may not have the resources to hire an engineering firm to go in and develop designs to inform them about ways to adapt to sea level rise. ECU itself is a very small engineering program. We have about 450 students total. And within that, we have 6 different concentrations. And that's important because on these teams I'm going to start talking about, we had a mixture of students. We had 2 environmental engineers, typically a mechanical engineer, and an industrial and systems engineer, all working on these capstone projects together. Our goal when we put this proposal to the NSF was to link the expertise of these engineering students with the communities in our region and hopefully help them advance more community-based design that fit the needs of the communities to help them adapt to sea level rise. We got the funding for this right before the COVID pandemic. And if you can imagine trying to incorporate student community engagement through Zoom in communities that don't have good broadband access, it was definitely a challenge. But the community we were working with said, we want to move forward. So that's what we did. So the area I'm working in is Lake Mattamuskeet. I'm sure most of you are familiar with that. It's circled there in green. And this is a sea level rise vulnerability map. The areas that are in darker red are more vulnerable to sea level rise. And you can see Lake Mattamuskeet is surrounded by dark red there. Give you an idea of the current status of the lake. Anytime the water level gets 1 foot above sea level, there's substantial flooding in the watershed. That's 1 foot above sea level is when we start to see substantial flooding. The bottom of the lake is below sea level. Here's my water quality piece. The lake is hypereutrophic and all the submerged aquatic vegetation in the lake has disappeared over the last 20 years. And that's a concern because it's a very important ecosystem for migratory waterfowl. And even folks that may not typically be concerned about water quality are, because it drives a lot of the economy there with both bird watchers and bird hunters coming to that area. So it's a very important ecosystem. I started working there around 2016, and Hurricane Matthew, if you remember, came through in 2016, and there was substantial flooding in this area. And that really kicked this off. U.S. Fish and Wildlife Service, North Carolina Wildlife Resources Commission, and Hyde County all came together and said, we need to do something. Also in 2016, Lake Mattamuskeet was added to the 303D list. So they said, let's get together, let's put together a watershed restoration plan. That was started in 2017. The North Carolina Coastal Federation led that effort. The plan was approved in August of 2019. And the current status is that all the partners have received some type of funding to try to design and implement solutions. So that's where we're at right now. The role that ECU played is when they started this, we didn't know how quick money would come in from other sources. And we wanted to start, at least keep the momentum going. We applied for a grant from the NSF and got funding to develop concept plans. Those of you that have worked with senior engineering students know that they're not going to produce something that can be implemented very quickly, but they can produce something that will give the community an idea of which direction do we want to go, which direction do we want to pursue. So we developed concept plans that were going to either reduce flooding or improve water quality in the lake. That's what we put in our proposal. We wanted to provide the community with cost estimates and effectiveness for whichever area we were focusing on with that project. As soon as we received funding, we were out in the community trying to get feedback. Which projects did we want to pursue? Which projects did we want to give the students? We got a lot of ideas, and we did our best for it to be community driven. In the end, most of the projects focused on flooding, very little water quality focus on those. Community engagement was a challenge. Instead of having the students out in the community as much as we like, unfortunately, they were sitting behind their computer screens at home because all the classes were done from home that year. So they were able to present at public and stakeholder meetings, but the participation of the stakeholders and the public in those meetings was much lower than it was pre-pandemic. So I'm just going to briefly mention the landowner project here. I'm not going to show any results from it. This was one that was chosen by the community because it could serve as a model for something that a single landowner could do on their property. Single project. This was actually the only project we did that was mentioned in the watershed restoration plan too. So that was interesting. But single project, not going to have a large impact at the watershed scale. But the folks the stakeholders were excited about this because it provides a model that can be implemented by individual farmers in the watershed. Other thing that was noted about this, and I'll point this out for the other projects, the design the students came up with should remain effective with increasing sea levels. Now, ever since I've been working down there, the members of the community have told me if we could just dredge the 4 canals that drain the lake, all our problems will be solved. I've heard that over and over. And that's one thing we heard when we started going out saying, what projects do you want us to evaluate? Is let's look at dredging the canals. Let's see what that does. 2017, I got a little bit of funding from ECU and was able to go out there and do a very crude survey of all of the canals and then go back through the documents in the courthouse and get an estimate of what were the original design dimensions of these canals. So back in 2017, I'm going to look specifically at Outfall Canal, which was the canal that was dug when the lake was drained 100 years ago. The primary canal there, it was dug to 10 feet deep. Now, 80% of that cross-sectional area for flow is gone. So you can imagine the flow potential for water moving from the lake out to the sound is much lower than it was when this canal was at its original dimensions. The other issue is there's tide gates on the lake that only allow water to flow out of the lake when the water level in the lake is greater than the sound. So that's also controlling flow. The students evaluated all the canals and their recommendation was dredge Outfall Canal only and dredge it to a depth of 8 feet, not the original 10-foot depth. They found that that extra dredging was not worth the additional cost. They estimated the total cost for this project between $4.5 and $6.75 million, plus additional design costs. And those of you that have worked in this area know that permitting is going to be a challenge for anything like this. So it's easily a $5 million project to get this going. So I'm going to show you some of the results of how we expect this project to perform. I'm going to show quite a few of these graphs, and this comes from Hurricane Matthew data in 2016. So kind of the worst case scenario that we have good data on. The blue line that's shown there is what happened. It's actual monitoring data from Lake Mesquite during Hurricane Matthew. I told you that 1 foot above sea level is when they start to see substantial flooding. That's shown by the red horizontal line there. During Hurricane Matthew, the lake spent 50 days above that threshold. If we simulated with a model, if that canal had been dredged out to what the students showed, we would reduce the peak water level by about 0.2 of a foot and reduce the duration of flooding from 50 days down to 21 days is what we expected. So that was something that the community members seemed to be excited about. This was a potential solution. Now, unfortunately, the students didn't have time to go in and evaluate what happens with sea level rise. So I've gone back in and added that into the model that we use. And with 0.6 of a foot of sea level rise, and depending on what projection you use, that could be 50 years from now, 100 years from now, but it's very, very likely to happen. Now our orange line is way above what happened during Hurricane Matthew. So this is not a long-term sustainable solution. This is something that with sea level rise, dredging the canals is not going to be effective. So now we've gone from a duration of flooding of 50 days to a duration of flooding of 80 days. So we've gotten worse. And really that comes down to the Pamlico sound levels higher, the water can't leave the lake. So another suggested project was, this is on the northeast corner of the lake, was moving water from Lake Mattamuskeet into the Mattamuskeet Drainage Association. The Mattamuskeet Association is around 42,000 acres, and they already have pump infrastructure there to maintain the water levels approximately 3 feet below sea level is where they maintain it currently. The students recommended adding a outflow control structure on an existing canal so that they could control how much water was flowing into the Mattamuskeet Association, direct that water to the northern end of the association where there's some existing surface water wetlands that have recently been restored, move the water through those wetlands before it's discharged either to the intercoastal waterway or a national wildlife refuge on the north side. So provide that opportunity for the water to be treated before it goes back out to sensitive bodies of water. To do this, they recommended installing two 48-inch pumps to move the water out of the canals on the association into the surface water wetlands. They estimated the cost for this project was going to be around $2.3 million. So let's look at how effective this would be. We see here that blue line hasn't changed. We still got the flooding duration of 50 days during Hurricane Matthew. We estimated that implementing this project would reduce the duration of flooding down to 15 days and substantially reduce the peak flood level there down to about 1.2 feet above sea level. So current conditions, this looked like this project would be fairly effective. Add in sea level rise and the water levels get a little higher. We've gone from a flooding duration of 15 days up to 20 days, but you can still see we've substantially reduced the amount of flooding. And the reason for the increase in flooding goes back to the fact that there's no natural drainage or very little natural drainage now because the tide gates can't open with the elevated Pamlico Sound. So this seems like a more viable long-term solution. The challenge is the funding part of it. So dredging the canal, yes, that's something that you would have to go and continually do more than likely, but it's not something that's going to be effective long-term. Typical model for funding something like this is everybody in the watershed contributes to the project being implemented and then the continuing operating and maintenance cost. About 75% of this watershed, the watershed's highlighted there or outlined there in red, about 75% of it is a national wildlife refuge. Now, I don't recognize anybody here in the audience being from Fish and Wildlife Service. I don't know if there's somebody online, but if you want to get the details of this, you may have to reach out to them. I'm going to do my best interpretation of what I have been told, but they cannot fund something long-term, the long-term operation and maintenance for something like this. They can contribute to the initial investment, the long-term operation and maintenance. My understanding, they cannot commit to that. So that's been a big discussion point within the watershed is, are we going to have 25% of the watershed funding the operation and maintenance for 100% of the watershed? And the landowners are very concerned about that. So that's one of the challenges that has come up over and over in the meetings that we didn't anticipate when we sent the students kind of into this environment, that they were going to be having to deal with that. So another thing that's come up, and this is from Raymond Smith, who's the industrial and systems engineer, is, okay, are there ways that we can increase maybe the initial cost of the system, but reduce those operation and maintenance costs using a more sustainable power source? So we're in the process now. We have a team seeing, can we develop a solar-based system to power these pumps? So then we can reduce the ongoing cost of having to provide, for example, diesel fuel to these pumps, the maintenance for the diesel engines. So we're looking at a more sustainable power source here. Now, don't ask me why they did this. These students went and took a pump capacity of 250,000 gallons per minute. The previous design only had a pump capacity of about 150,000 gallons per minute. So I'm not sure why they used the bigger pumps, but they did. They estimated that they used enough 300-watt solar panels to cover about 1/4 of an acre, added a 16 megawatt hour battery system that they could operate these 250,000 gallon per minute pumps continuously for 15 days to pump the water out of the lake. They don't have a complete cost estimate at this point, but just an initial guess that they were willing to give me was it would easily double the cost of a project, any of the projects that I've mentioned. So it's a substantial cost that would incur with that. We haven't completely run all these scenarios yet, but here's some initial results. This is a much longer time period than I've shown before, but we still, if you go look at October 2016, that's when Hurricane Matthew went through, you can see the increase in pump size here is actually even more reduce the severity of flooding that they would experience there. So we would expect this project to work. The interesting thing to add in, and this is something we still have to go back and work through with the students, is the duration the pump's running. What's shown there in the green is when the pumps would run in these scenarios. If you look at it closely, you'll see that 2017 and 2018, those pumps would never cut on. So we would have 2 years where the pumps were never even needed under current conditions. But the periods where they are on are typically longer than those 5 days. So there would have to be some additional power pulled from the grid to make this system work. So just a quick summary of where we're at with this project. Canal dredging is still supported by many in the community. And that's something that even that's being pursued now. They're still investigating what it would take to go in and dredge the canals, even though they understand it's not necessarily a long-term solution. If they're going to have a long-term solution, I think everyone's starting to recognize that pumps are going to be involved somehow. And I have heard a lot more discussion recently about how do we fund that? How do we make that work, a model of that work, where maybe the Fish and Wildlife Service that has 75% of the watershed can contribute. Solar microgrid system may be a way of reducing the operation and maintenance costs for pumps there. And here's my last little bit of water quality. I didn't include this in the presentation, but the students did evaluate this. Surface flow wetlands that are available within the watershed are being restored through other funding sources are a great way of improving the water quality when it's being pumped into these surface water wetlands. Great opportunity to improve the water quality before it's discharged to places like the Pamlico Sound or the Intercoastal Waterway. So thank y'all for sticking around, even though this wasn't really a water quality presentation. And now I'll take any questions y'all have.
Thanks very much, Randall. Does anyone have a question for Randall?
Yeah, Randall, not to be a wet blanket or anything, but at current rates of the sea level rise, how long is it before Mattamuskeet becomes part of Pamlico Sound?
So that is something that I don't have the exact answer to. And it really depends on how high they're willing to build the berms around it. And that's something that for example, the Mattamuskeet Association is using, I believe, FEMA funds right now to increase the berm height around that whole association. And that, I'm guessing you're getting at the idea of retreat with that question a little bit. And that is something that you don't hear mentioned in the community. It's all, how can we maintain our current lifestyle and adapt to this? So, but yes, I think everybody involved with the project is realistic that eventually there's a great chance that the ecosystem we're working on is just eventually part of the Pamlico Sound.
Thanks so much for the presentation. I'm curious, with the water in Lake Mattamuskeet, I think you said it's really eutrophic. And I'm wondering, I could see if you dredge the canal, you'd have a lot of that water carrying lots of nutrients going into wherever it's discharging. But even with the pumps, even if you're putting it into a natural wetland, that you probably need a discharge permit for that water. And have you all looked at the concentration of nutrients and whether that would be an issue?
So I can't speak to the permitting side of that. That was one of the things we did pay for in this project, is we hired a consulting professional engineer to come in and review some of that. And the the discharge of nutrients was not something that came up in that. There's a lot of other things other hurdles that would have to go through first. I can tell you the first hurdle, especially with this project, is I'm not sure how many of y'all are familiar with the interbasin divide. And moving water between basins is an issue. Well, if I'm not mistaken, there's an interbasin line that's drawn right across the Mattamuskeet Association that we would be moving water across if something like this were implemented. So that was the biggest thing that came up from that perspective when we're talking about permitting. I know that there was concern, especially on the north side of this, the Alligator River National Wildlife Refuge, and there's concern from them of, are we going to be, if this project were to ever be implemented, moving nutrients into what's considered more of a pristine habitat there? So yes, that's definitely a concern, but I don't have, to answer your question, I think I'm answering it. I don't know the details on the permitting side for that.
Well, thanks so much, Randall. Thanks everyone so much. We're just out of time right now. Time for a break. And if you have any other questions for the speakers, I'm sure they'll be glad to catch you at the break. Thanks for a great session.